History of Life on Earth. Chapter 6, Invertebrates. 100 Ways to Build an Animal. 6.1 The Hole That Became Your Anus. I'm sorry, there is no dignified way to open this chapter. Go back to Chapter 5, to the fold, that dimple and the hollow ball of cells that you once were, pressing inward to make a body. The dimple is called the blastopore, and it is the first opening any animal has. One hole. A single doorway between the inside and the outside. Eventually, a body needs two. Food has to come in somewhere, and waste has to leave somewhere else. So the embryo punches a second hole through to the other end of the tube, and now there is a mouth and an anus and a one-way road between them. The question is, which hole was there first? And the animal kingdom, it turns out, is split down the middle by the answer. In the Protostomes, from the Greek for mouth first, the original blastopore becomes the mouth. The anus is the afterthought, drilled through later at the far end. In the Deuterostomes, mouth second, the original blastopore becomes the anus. The mouth is the afterthought. You are a Deuterostome. That is not a joke, and it is not an exaggeration. The very first opening your body ever made, the first structural decision of your entire existence, the primal doorway that started your architecture, became your anus. Your mouth is a later addition, a second thought, drilled in from the other side afterward. An insect did it the other way around. So did a snail, a squid, a lobster, a worm, and the overwhelming majority of animals on Earth. So this chapter is about a family reunion, and it's worth knowing where you sit at the table. Everything that follows, the crabs and the octopuses and the earthworms and the sea stars, is your relative. But the seating chart is not the one your intuition would draw. And it begins with a hole. 6.2, a word that isn't real. Before we go further, I have to take a word away from you. Invertebrate is not a real category. It is defined entirely by an absence, anything without a backbone, which is exactly as biologically meaningful as sorting the world's animals into elephants and non-elephants. A group defined by what it lacks is not a family. It is a leftover. Biologists call this a paraphyletic group. A group that contains some of an ancestor's descendants, but not all of them. And here is why that matters enormously for this chapter. A sea star is an invertebrate. So is a lobster. But a sea star is more closely related to you than it is to a lobster. Read that again because it is genuinely one of the most counterintuitive facts in zoology. Sea stars are Deuterostomes. So are sea urchins, sea cucumbers, and a group of unlovely burrowing creatures called acorn worms. So are you. In the deep architecture of development, in the hole that came first, a sea urchin and a human being made the same choice, and a beetle made the other one. If you were drawing the true family tree of animals, you would not put a line between vertebrates and everything else. You would put the line where evolution actually put it, between the mouth-first animals and the mouth-second animals, more than 500 million years ago. So invertebrate is a word of convenience, and I'll keep using it, because everyone does. But hold it loosely. It doesn't carve nature at the joints. It carves nature at the spine, which is not the same thing at all. It just happens to be the bone we care most about, because it's ours. 6.3 The split and the surprise. The mouth first, mouth second divide comes with a whole package of other early developmental differences. And for most of the 20th century, this package was the map of the animal kingdom. Protostomes typically show spiral cleavage. The first few cell divisions come off at an angle, so the cells stack in a twist, like a spiral staircase. And their development is typically determinate. Each cell's fate is locked in almost immediately. Remove one cell from a 4-cell protostome embryo, and you don't get a smaller animal. You get an animal missing a quarter of itself, permanently, because that cell was already committed. Deuterostomes typically show radial cleavage, cells stack squarely, directly on top of one another. And development is typically indeterminate. The early cells keep their options open. Split a Deuterostome embryo in half early on, and in many lineages, each half can build a complete animal. Which is worth pausing on, because it's the reason identical twins exist. Your particular Deuterostome flexibility, the fact that an early human embryo can be cut in two and produce two whole people, is a direct inheritance from the same ancient developmental style that lets a sea urchin embryo be halved and grow two urchins. Twinning is a Deuterostome trick. For 100 years, this was the tidy picture. Then, in the late 1990s, molecular biology arrived and knocked it over. Researchers began comparing gene sequences directly, reading the ribosomal RNA of one phylum against another, and the tree that came out did not match the tree drawn from anatomy. The protostomes, it turned out, are not one group but two, and the boundary between them is not where anyone expected. Ecdysozoa, the molting animals. What unites them is not how their embryos cleave, but a single brutal fact of life. They are trapped inside a rigid cuticle, and to grow they must periodically shed the whole thing. Arthropods are here, insects, crustaceans, spiders. And so, astonishingly, are the nematodes, roundworms. Soft, unsegmented, legless, eyeless, blind little threads that look nothing whatsoever like a crab. Tracheae, a mouthful of a name, also called Spiralia, covering the mollusks, the annelid worms, and the flatworms, among others. And here is the surprise that shook the field. Earthworms and centipedes are both obviously, conspicuously, gorgeously segmented, built from a row of repeating units, like a train of identical cars. Anyone can see it. For most of a century, biologists grouped them together on exactly that basis, in a class called Articulata, and it seemed like one of the safest calls in zoology. The molecules say no. A centipede is more closely related to a roundworm and unsegmented thread than to an earthworm. Segmentation, the most obvious anatomical feature in the room, was leading everyone astray. Which leaves a question still unresolved. Did arthropods and annelids both inherit segmentation from a shared segmented ancestor, and the nematodes lost it? Or did arthropods and annelids invent segmentation twice, separately, because it's simply a very good idea? We don't know. But you should recognize the shape of the question by now, because this book keeps running into it. Life finds the same answers again and again, and it is often maddeningly difficult to tell an inheritance from a coincidence. 6.4 Growing Ahead. Chapter 5 left the nervous system as a nerve net, the diffuse mesh of a jellyfish, with no center, because a radially symmetric animal has no front to speak of. Bilateral symmetry destroys that logic completely. An animal with a left and a right end, a front and a back has a direction of travel. It goes one way. And that means one particular end of it is always the end that meets the world first, that arrives at the food, the mate, the predator, the wall, before any other part of the body knows they exist. The consequences are immediate and irresistible. Put your sensors at the leading end. That's where the information is. And then put your processing right behind the sensors, because a signal that has to travel the length of the body before anything can decide what to do about it as a signal that arrives too late. This is cephalization, the evolution of a head. And a head is not merely a body part. A head is an argument that says the future arrives from a particular direction, and it pays to look at it. Every animal that hunts, flees, or explores has one. Where the resulting nerve cord runs, though, splits the animal kingdom along the same fault line as the blastopore. Protostomes run their main nerve cord along the belly, beneath the gut. Often it's paired, a double cord, with swellings of nervous tissue, ganglia at each segment, joined by cross connections into a shape that looks, under a microscope, exactly like a rope ladder running down the underside of the animal. Deuterostomes, chordates specifically, run their nerve cord along the back above the gut. And it isn't solid. It's a hollow tube. Your spinal cord is on the wrong side of your body compared to an insect's, and there's a hole down the middle of it. Which brings us to one of the strangest stories in the history of anatomy. In the 1820s, the French naturalist Étienne Geoffroy Saint-Hilaire stood up and proposed that a vertebrate is essentially an arthropod turned upside down. Flip a lobster over, he said, and the nerve cord that was beneath the gut is now above it. Flip a lobster, and you have the layout of a vertebrate. The idea was regarded as absurd. Geoffroy's great rival Georges Cuvier, the most powerful anatomist in Europe, demolished him in a celebrated public debate in 1830. And the notion of the upside-down lobster passed into science's long catalog of colorful nonsense, where it sat for 160 years. Then, in the 1990s, developmental geneticists found the genes that tell an embryo which side is the back and which side is the belly. In a fruit fly, a gene called decapentaplegic says back, and a gene called short gastrulation says belly. In a frog, the very same genes, the direct, unmistakable evolutionary counterparts, still recognizable in sequence, are both present and both doing the same job. Except they are swapped. In the vertebrate, the fly's back gene specifies the belly, and the fly's belly gene specifies the back. The molecular map of the body is the same map. It has simply been turned over. Geoffroy St. Hilaire, ridiculed and forgotten, appears to have been looking at something real. The most economical reading of the evidence is that the last common ancestor of flies and frogs already had a centralized nerve cord running down one side of its body, and that's somewhere in the deep Precambrian. One lineage rolled over, possibly to swim on its back, possibly to burrow. Nobody knows. Whatever the reason, if the hypothesis holds, then somewhere in the ancient mud, an animal flipped, and every animal with a backbone has been walking around upside down ever since. It is still debated. But it is a beautiful thing to think about while looking at a lobster. 6.5 Plumbing. An animal that gets thick can no longer rely on diffusion. And Chapter 5 explained why. Diffusion is fast over a distance of microns and hopeless over a distance of millimeters. Get big and you must build plumbing. There are two ways to do it, and the difference between them sets a hard ceiling on what an animal can become. Open circulation, the arthropod and most mollusk solution, is refreshingly casual about the whole thing. A heart pumps fluid, called hemolymph, out into the body cavity, where it simply sloshes around the organs, bathing them directly. Eventually it works its way back to the heart through little valved openings, and gets pumped out again. There are no capillaries. There is barely a circuit. Your blood is confined to vessels for its entire journey. An insect's is not, and if you opened an insect you would not find blood in anything. You would find the insect swimming in it. It's cheap, it's simple, and it works fine, as long as you stay small and don't ask too much of it. But the pressure is low, and the delivery is imprecise. You cannot push fluid hard through a puddle. Closed circulation, annelids, vertebrates, confines blood entirely within vessels, arteries, capillaries, veins, a sealed circuit under real pressure, which means you can direct it. You can send more blood to a working muscle and less to a resting gut. You can raise the pressure and drive it fast. You can be large and thick and demanding and sustain it. And now for the twist, which is one of the best stories in animal evolution. The mollusks are open circulation animals. Snails, open. Clams, open. It's the family standard, except for the cephalopods. Squid and octopuses are mollusks, genuine, card-carrying relatives of the garden snail, and they abandoned the family plumbing entirely and evolved a closed circulatory system, independently, from scratch. Because they became fast active predators, and the old system could not keep up. They also have 3 hearts. 2 of them are branchial hearts, one at the base of each gill, whose only job is to force blood through the gills. The third is a systemic heart that drives the oxygenated blood out to the body. 2 pumps to load the fuel, 1 to deliver it, and when an octopus swims by jetting water, the systemic heart actually stops beating, which is exhausting. It's one of the reasons octopuses prefer to walk. Their blood isn't even red. It carries oxygen on a copper-based protein rather than an iron-based one, and it runs blue. Look at what has happened here. Faced with the same problem, an active predatory life, a body that needs oxygen delivered hard and fast, a mollusk and a vertebrate, separated by 600 million years, arrived at the same answer independently. Watch for this. It is about to happen again, in the same animal, with something much more startling than a heart. 6.6 skeletons, and the terrible cost of armor. A skeleton does 3 jobs at once. It holds the body up, it keeps predators out, and, most importantly, and least obviously, it gives the muscle something to pull against. A muscle can only contract. It cannot push. Without something rigid or incompressible to work against, contraction achieves nothing at all. Bilaterians found 3 answers. Water. Chapter 5 introduced the hydrostatic skeleton, and the earthworm is its master. Fluid can't be compressed. Squeeze a segment with circular muscles, and it must get longer. Squeeze it with longitudinal muscles, and it must get shorter and fatter. Run those contractions down the body in a wave, segment by segment, anchoring with bristles as you go, and you have peristaltic crawling, a worm boring through packed soil using nothing but muscle and water, and generating serious force while doing it. Nematodes, which have no circular muscles at all, can only shorten one side against the other, which is why roundworm doesn't crawl, it thrashes, whipping side to side, and the frantic C-shaped lashing you'd recognize if you've ever seen one under a microscope. Same principle. Half the equipment. Very different animal. Armor. The arthropod solution is the exoskeleton, a rigid external cuticle of chitin and protein, secreted by the skin beneath it, with the muscles anchored to its inner surface. The animal is a suit of armor with the meat on the inside. It is, without exaggeration, one of the most successful structures in the history of life. It protects. It provides leverage. It can be shaped into a jaw, a claw, a lens, a wing. And, critically, because it is waterproof, it lets an animal walk out of the ocean without drying out. The exoskeleton is a large part of why arthropods conquered the land while nearly everything else was still stuck in the water. But it has one catastrophic flaw. It cannot grow. A rigid box cannot expand. So the animal inside it has to get out. Ecdysis, molting, is what gives the ictyosazoa their name. And it is the most dangerous thing an arthropod ever does. It secretes a new, soft, wrinkled cuticle beneath the old one. It dissolves the inner layer of the old shell. And then it splits itself open and drags itself out, every leg, every antenna, every mouthpart. And, in insects, the lining of the breathing tubes, and the gut, pulled free like a hand out of a glove. Then it swells with air or water to stretch the new cuticle before it sets. And waits, soft and helpless, unable to run, unable to fight, sometimes barely able to stand while its armor hardens over hours or days. Every arthropod on Earth must do this repeatedly to grow up. Many of them die doing it. Stuck, exhausted, deformed, or simply eaten while soft. That is the trade, where the armor becomes a prison you have to break out of, over and over, and each escape can kill you. The third answer, a skeleton built inside the body, out of living tissue that grows with you, is coming. It shows up first, quietly, in the sea stars at the end of this chapter, and it will change everything in the next one. 6.7 Air. Getting oxygen is the same problem as circulation, one step earlier. You need surface area, and you need it in contact with the outside world. Gills are the aquatic answer, found across the mollusks, and worms, feathery outfoldings of tissue, thin-walled and richly supplied with blood, waving in the water. In mollusks they're called ctenidia, and many of them multitask beautifully, the same beating cilia, that pull a current of water across the gill for oxygen also filter food particles out of it, breathing and eating on a single stroke. But gills are useless in air. Without water to hold them apart they collapse into a wet clump, which is precisely why a fish suffocates on a dock while drowning in oxygen. So the animals that came ashore had to reinvent breathing. The insect solution is so unlike ours that it takes a moment to absorb. Insects do not carry oxygen in their blood at all. Instead, an insect's body is riddled with a branching network of tubes, tracheae, splitting into finer and finer tracheoles, that carry air itself, as gas, directly to the tissues. The tubes end in contact with individual cells. Oxygen is delivered to your muscles by a fluid, pumped by a heart, through a vessel. Oxygen is delivered to a beetle's muscles, by air, arriving down a pipe, straight from the sky. The pipes open to the outside through valve holes down the insect's flanks, called spiracles, which can be clamped shut to stop water escaping. The elegance of this is easy to miss. It decouples breathing from circulation entirely. An insect's hemolymph doesn't have to carry oxygen, which frees it to do nothing but distribute food and collect waste, and it means an insect flight muscle, working harder per gram than almost any tissue in the animal kingdom, gets its oxygen delivered directly to the door, with no transport delay at all. But there is a price, and it is why the world is not full of enormous insects. Air moves through those tubes largely by diffusion, and diffusion, once again, is only fast over short distances. Make the insect bigger and the tubes get longer and the oxygen delivery gets worse, until it fails. The tracheal system sets a ceiling on body size, and the ceiling is quite low, except when it isn't. In the Carboniferous, 300 million years ago, atmospheric oxygen rose far above modern levels, and in that thicker, richer air, the ceiling lifted, and dragonflies grew wings that spanned 2.5 feet, and centipede relatives grew to the length of a car. When the oxygen fell, they went with it. Spiders and scorpions found a different answer. The book lung, a chamber inside the body containing a stack of thin tissue plates, layered like the pages of a book, with air in the gaps between them. It's a gill turned inside out, folded up, and tucked into a pocket where it won't dry out. And that is quite literally what it is. Book lungs appear to be modified gills, the flattened, plate-like gill structures of an ancestral marine chelicerate, retained and repurposed for air. Biologists call this an exaptation, a structure that evolved for one purpose being co-opted for another entirely. You will meet exaptation constantly, for the rest of this book. Feathers before flight, jaws from bill arches, lungs from swim bladders, or, as it turns out, the other way round. Evolution has no drawing board. It only has whatever is already on the bench. 6.8 The gift of the second hole. Back to where the chapter started, because that second opening is not a plumbing detail. It's a revolution. A cnidarian has one hole. Food goes in, waste comes out the same way, which means the gut is a single chamber that has to do everything in sequence, one batch at a time, and it has to be flushed before the next meal can come in. Traffic in both directions, through one door. Most bilaterians instead have a complete gut, a tube, mouth at one end, anus at the other, one-way traffic. The obvious one is logistical. You can eat again before you finish digesting the last meal. Food and waste never meet. But the real prize is subtler, and far more important. A one-way tube can be specialized along its length. Think about what that means. In a one-hole gut, every part of the chamber has to be a generalist, because everything that happens there. But in a tube, food moves through in a predictable direction, which means each stretch of the tube can commit to a single job, tuned precisely for it, knowing exactly what condition the food will be in when it arrives. So a region up front can be built for grinding. The next region can bathe the food in acid, at a pH that would destroy the tissues on either side. The next can neutralize the acid and pour in enzymes. The next can be folded and refolded into an enormous absorptive surface for pulling nutrients across. And the last can specialize in wringing the water back out of what remains. Foregut, midgut, hindgut. An assembly line, invented in a worm, elaborated in every animal sense, and running in you right now. The second hole was not an indignity. It was the beginning of the factory. 6.9 The Arthropods. Life's most successful idea. If you want to know what animal life on Earth actually looks like, not the mammals and birds cartoon of a nature documentary, but the real distribution, the answer is arthropods. Insects, crustaceans, arachnids, myriapods. Over 1 million species described. And credible estimates of the true total running to 5 or 10 million or more. The great majority of animal species that have ever been named are arthropods. Roughly 1 in 4 of all named species of any kind is a beetle. A famous story, probably improved in the retelling. The biologist J.B.S. Haldane was asked what the study of nature had taught him about the Creator, and he is said to have replied that God appears to have an inordinate fondness for beetles. What makes the body plan so successful comes down to 2 ideas, and they're really one idea applied twice. Jointed appendages. Arthropod means jointed foot. The ancestral arthropod had a long row of segments, each with a pair of simple limbs, essentially the same limb, over and over down the body. And then evolution began modifying copies. Take that basic repeated limb, and reshape it and you get an antenna, a jaw, a fang, a claw, a walking leg, a swimming paddle, a feeler, a pincer, a structure for holding eggs, a structure for holding a mate, a wing. Every one of those is the same ancestral appendage, edited. A crab's claw and a butterfly's tongue, and a spider's fang are all the same body part, elaborated in different directions. This is the Evo-Devo principle from Chapter 5, Made Flesh. You do not need new genes to make a new tool. You need a repeated structure, and a switch that says make this one different. Segmentation. The same logic, applied to segments instead of limbs. Take a long uniform row of segments, and fuse groups of them into functional regions. A head for sensing and eating, a thorax for locomotion, an abdomen for digestion and reproduction. Head, thorax, abdomen in an insect, cephalothorax, an abdomen in a spider. Specialization by neighborhood. And then, in the Devonian, an insect did something no animal had ever done. It flew. Insects invented powered flight. Not birds, not bats, not pterosaurs. Insects, perhaps 350 to 400 million years ago. Tens of millions of years before any vertebrate got off the ground, and hundreds of millions before a bird existed. For an immense stretch of time, if something was flying over your head, it was an insect, and there was nothing else up there at all. And one more metamorphosis. A caterpillar and a butterfly are the same animal, and they share almost nothing. Not their diet, not their habitat, not their body, not their behavior. That is the point. Metamorphosis lets a single species split its life into 2 entirely different ecological careers, so that the young are not competing with their own parents for the same food. It is a way of being 2 animals with 1 genome, armor, modular limbs, modular bodies, flight, and 2 lives per lifetime. That's how you take over a planet. 6.10 The mollusks, and the mind in the water. Mollusks are built from 4 ingredients. A muscular foot, a visceral mass holding the organs, a mantle, a fold of tissue that secretes the shell and encloses a cavity, where the gills sit, and a radula, a rasping tongue studded with rows of tiny teeth, used to scrape food off surfaces. That's the kit. Now watch what gets built from it. A snail puts the foot underneath and crawls on it, hauls the shell on its back, and grazes with the radula. A clam flattens the whole body between 2 shells, gives up on having a head entirely. No head, no radula, no brain to speak of, and just filters water through its gills, forever. And then there are the cephalopods, which took the foot, split it into arms, put them around the mouth, threw away the shell, and became the most intelligent invertebrates on Earth. We've already met their 3 hearts and their closed circulation. But that isn't the astonishing part. The eye. A cephalopod eye is a camera eye. It has a cornea, an iris, a lens, and a retina. It focuses an image. It is, in overall function and general architecture, remarkably like your eye, and it is not inherited from any common ancestor. The last common ancestor of you in a squid was a small, simple, worm-like thing in the Precambrian that had, at most, a patch of light-sensitive cells. It did not have an eye. Nothing on the path from it to the octopus, and nothing on the path from it to you, shares an eye. 2 lineages, 600 million years apart, independently invented the camera eye, because, when the problem is form an image with light, there are only so many ways physics will allow you to solve it. Optics is optics. Given a light-sensitive surface, an aperture, and an animal that needs to see, the camera is where evolution ends up, whether it's building a vertebrate or a mollusk. And now the detail that gives it all away. Your retina is installed backward. The photoreceptors, the cells that actually detect light, face away from the incoming light. The nerve fibers and blood vessels that serve them run in front of them, between the light and the sensors. So every photon that reaches your photoreceptors has to pass through a layer of wiring first. And those wires have to exit somewhere. They gather into a bundle and punch straight through the retina to get out to the brain, leaving a hole in your visual field with no receptors at all. That is your blind spot. You have 1 in each eye. Your brain edits them out so seamlessly that most people go their whole lives without noticing. The octopus retina is installed the right way round. The photoreceptors face the light. The wiring runs out the back, where it belongs. The octopus has no blind spot. This is the fingerprint that proves the 2 eyes are independent inventions. If we had inherited the camera eye from a shared ancestor, we'd share the wiring. We don't. Evolution built the same instrument twice, and it happened to make a sloppy wiring choice in one lineage and a clean one in the other, and neither can go back and fix it, because you cannot rewire a working eye halfway through. Every animal is a museum of decisions that can no longer be revisited. And the cephalopod brain. Octopuses solve puzzles, open jars, use coconut shells as portable shelters, recognize individual human faces, and escape from tanks with a persistence that has driven aquarium staff to despair. Most of their neurons are not in the central brain at all but distributed down the arms, which have a substantial degree of independent processing, so that an octopus arm can explore, taste, and make decisions with a considerable amount of local autonomy. This is a mind built on an entirely separate plan from ours, in a body with no bones. Descended from a snail, it is the closest thing to an alien intelligence we are ever likely to meet, and it has been living in the ocean the whole time, watching us. 6.11 The Annelids. The beauty of repetition. Annelids, earthworms, marine bristleworms, leeches, perfected an idea that will matter enormously in the next chapter, segmentation, or metamerism. An annelid is built from a row of near-identical modules. Each one has its own coelomic compartment, walled off from its neighbors by internal partitions. Its own muscles, its own ganglion, hanging off the main nerve cord, its own pair of excretory organs, the nephridia, filtering waste from its own little chunk of body. The animal is not a single machine. It's a sequence of machines, running in parallel, coordinated, but semi-independent. The advantage is compound, independent control. Because each segment is walled off, the worm can contract muscles in one segment, while relaxing them in another, which is exactly what peristaltic crawling requires. Without the partitions, squeezing one end would just bulge the other, and the animal would flop uselessly. Redundancy. Damage one segment and the rest carry on. Many annelids can regenerate lost segments entirely. When a body is a row of modules, losing one is a repair job, not a catastrophe. Scalability. And this is the deep one. To make a bigger annelid, You do not need to redesign the animal. You just add more segments. The developmental program that makes a segment is already written. Run it more times. Evolution can change an animal's size without changing its architecture at all. That is an extraordinarily powerful thing to have in a body plan. And it is why segmentation keeps appearing in the most successful animals on Earth. Arthropods are segmented. Annelids are segmented. And so are you. Not obviously, not on the outside. But your vertebrae come in a row. Your ribs come in a row. The muscles between your ribs come in a row, each with its own nerve, and its own artery running out from the spinal cord at its own level, in perfect series, exactly like the ganglia and nephridia of an earthworm. Peel back the skin, and you are a stack of modules with a head on top. Chapter 7 will show you where they come from. 6.12 The Echinoderms, our strangest relatives. And now, at last, back across the Great Divide, to the Deuterostomes, and to us. The Echinoderms, sea stars, sea urchins, brittle stars, sea cucumbers, sea lilies are your cousins. They are on your side of the family. And they are, without competition, the weirdest animals in this book. Here is the first oddity. An echinoderm larva is bilaterally symmetric, a small, sensible, free-swimming thing with a left and a right, exactly as a deuterostome should be. And then it undergoes a metamorphosis so violent and so total that it essentially rebuilds itself from the inside out, and emerges with five-fold radial symmetry. 5. Not 2. A sea star has no left and right. It has no front. It has 5 of everything, arranged around a center. And it can move in any of 5 directions with equal facility. And it does not care which. Bilateral symmetry was one of the great inventions of Chapter 5, the foundation of heads, brains, and directed movement. The echinoderms threw it away. They had it, in the larva, and they abandoned it as adults, because they had settled into a slow, grazing, seafloor life where facing every direction at once is more useful than facing one. They gave up the head, deliberately, which should tell you something about how ruthlessly indifferent evolution is to the things we consider progress. Then there's the way they move. Echinoderms have no legs, no fins, no cilia to speak of. They have a water vascular system, an internal network of canals filled with seawater, drawn in through a sieve-like plate on the animal's surface called the madrepride, distributed around a ring, canal, and out along 5 radial canals. And branching off those canals are hundreds, sometimes thousands, of tube feet, small, soft, hollow, extendable projections that emerge through the underside of the animal. Each one is paired with a muscular bulb. Squeeze the bulb, and fluid is forced into the tube foot, and the foot extends. Relax it, and the foot pulls back. The tip can grip. A sea star walks by hydraulics. It is a machine that moves by pumping seawater through a plumbing system into thousands of tiny hydraulic pistons, coordinated well enough to carry the animal across the seabed, up a rock face, and, famously, patiently, and with a force that can be measured in kilograms, to pull open the shell of a mussel that is holding itself shut for its life. There is nothing else like it anywhere in the animal kingdom. It is a genuinely alien technology, and it evolved in our own family. And finally, the thing that makes echinoderms belong in this book's argument. Look at what their skeleton is made of, and, more importantly, where it is. An echinoderm skeleton is built from calcium carbonate plates, called ossicles. But they are not a shell. They are not a cuticle. They are not on the outside. They are embedded within the body, inside the skin, covered by living tissue. And they are built from mesoderm, the middle germ layer, the same layer that will build the bones in your arm. This is an endoskeleton, a skeleton inside the animal, made of living tissue, growing continuously with the body that carries it. Do you see what that solves? The arthropod is trapped in its armor and must tear itself out to grow. The echinoderm skeleton grows with it. No molting, no helpless soft days, no armor to escape from. The skeleton is not a box the animal lives in. It is part of the animal. The arthropods built the greatest suit of armor in the history of life, and it capped them. It made them small, and forced them, over and over, to crawl out of their own bodies and lie there hoping nothing found them. Their deuterostome cousins, on the other side of the family, quietly did something else. They put the skeleton inside, and that decision, living bone, growing with the animal, with no ceiling on how large it can become, is the reason there are whales. 6.13 The Threshold. Look at what the Bilaterian body plan has produced. 1 million species of jointed, armored animals with modular limbs, some of them flying, most of them beetles. A lineage of shelled grazers that produced, in one branch, a camera eye, 3 hearts, blue blood, and a mind. Worms built from repeating modules, boring through the earth on nothing but muscle and water. and a group of star-shaped hydraulic machines that gave up having a head. Every one of them solving the same handful of problems. Hold the body up, move it, feed it, oxygenate it, coordinate it, and arriving at wildly, gloriously different answers. And running through all of it, the patterns this book keeps returning to. Convergence, the camera I invented twice. Closed circulation invented twice. Segmentation possibly invented twice. Exaptation, the gill that became a book lung. Redeployment, the same ancestral limb, edited into a claw, a jaw, an antenna, a wing. Now hold two facts side by side. The Echinoderms are Deuterostomes, and they put their skeleton inside their bodies, built from mesoderm. And somewhere in the same Cambrian seas, another Deuterostome lineage. Small, soft, easy to overlook. No armor and no arms and no hydraulics. Was swimming with a stiff flexible rod running the length of its back. A hollow nerve cord above it. A row of slits in its throat. And a tail that continued past its anus. Nobody would have picked it. It had none of the advantages. Against a meter-long Anomalocaris it was food. And against a trilobite it was defenseless. And against an octopus it would not have been very impressive company. But it had a spine and embryo, and it had the Deuterostome habit of building its skeleton from the inside. And in that combination was every fish that has ever swum, every amphibian that crawled onto a mud bank, every reptile, every bird, every mammal, every whale and every elephant and every hand that has ever held a pen and written any of this down. That is chapter 7.